Hi guys, this is Zachary Bowie and this is my teratogen presentation. Teratogen is basically anything that affects the fetus while the mother is pregnant, like a chemical or an infection that causes developmental abnormalities. So I'm actually going to be talking about congenital toxoplasmosis, which is a syndrome that's caused from when the mother gets infected with the Toxoplasma gondii parasite. So this parasite is actually one of the infamous members of the TORCH group of congenital infections. Those include toxoplasmosis, rubella, cytomegalovirus, herpes simplex virus, and now Zika. So the O for TORCH used to be part of toxoplasma for the TO. Now it's actually stands for others, which would include that Zika virus. So about toxoplasma in particular, it is an obligate intracellular parasite. So it only gets into all nucleated cells and can cause infection. And I'll talk about how that causes infection in just a moment. But the big picture is it invades the cell, replicates inside, and then it bursts out of the cell, causing destruction. So once it does get inside the cell, it forms this, you can see here, this vacuole that gives it a protective membrane from the rest of the cell. And this allows it to have replication to be unhindered. One of the main proteins that allows it to get inside the cell and also wreaks havoc in other ways that I'll talk about is this rhoptry or ROP protein. So it's actually secreted from the parasite itself, and it gets onto the vacuole membrane. And then once that parasite and all of the daughter parasites, once they all leave the membrane, they actually release that ROP protein. And I'll talk to you in a minute about what that ROP protein does. There's actually two ways that the parasite can cause damage to the cell. One of them is, like I said, is of course lysis of the cell as it's exiting. It replicates inside that vacuole until so many are in there and it just bursts, and then it bursts the cell itself. So the other way is indirect killing by an inflammatory response. So this is mediated a lot by IgA and eosinophils that attack parasites. So congenital toxoplasmosis itself, like I said, this is the syndrome that it causes. The reason I show a cat here is if you've never heard of this, toxoplasmosis actually is found in the intestines of domestic cats. They don't necessarily cause infection, but it can be excreted in their stool. So that's why they tell pregnant women not to change litter boxes, because they could contract this disease. So it's not just the pregnant mothers that are at risk, it's also immunocompromised patients. Most people, including those of reproductive age in women, most people have a strong enough immune system to actually fight this parasite off pretty quickly. So once the mother does get infected, this can cause a lot of issues with the developing fetus. And most of the mechanisms of this are not currently known, but I will talk about one that was just recently found. So kind of in descending order here are the bad outcomes that can happen. Abortion, of course, is on the more severe side. Neural malformations are one of the most common ones that we see in live births. And then that's followed by intracranial calcifications and other issues such as liver enlargement and spleen enlargement. So the severity and which one of these malformations occurs kind of depends on timing. If it occurs during implantation, abortion could happen, or if it's during the time of the neural cord formation, of course, there could be a lot of spinal cord and brain issues. And you can see here that the incidence is 1.5 per 1,000 live births. So going into one of the most common issues of live births, these are the neural manifestations of the infection. So this is actually an MRI of a fetus at 29 weeks, which you would think around this time they would have a much smaller ventricles. But you can see here that there's significantly less brain tissue and a lot more fluid in this area. So how does this happen? First, I'll give you some background information. Neural stem cells are one of the cells that the parasite actually enters, and I'll tell you about how it gets in just a moment. But these neural stem cells are what give rise to the lining of the neural tube, and they also help scaffold for the migration of other progenitors into that area and help differentiate into different glial cells. And the way these neural stem cells do this, they rely heavily on Wnt signaling, and this helps with proliferation and differentiation into the neurons. And I show you here a figure of the actual Wnt signaling process, and I'll talk about it more at the end and how it relates to the parasite infection. But essentially, you can see here the Wnt ligand lands on the frizzled receptor, which allows beta-catenin to be released and cause transcription of different Wnt target genes. So how does the infection actually cause issues? Well, it actually inhibits that Wnt beta-catenin signaling pathway. And the way it does this is through the virulence factor ROP18. And this can cause abnormal proliferation, differentiation, or apoptosis. So to kind of outline this, I'm going to actually talk about this study done by Zhang and others about using this ROP18 protein from the parasite Toxoplasma gondii and how that actually inhibits the Wnt pathway. So their hypothesis, as I just said, was that this protein is secreted from the parasite, and you can see the parasite here. It's actually using some of these proteins to get inside the cell. But once it gets inside, it actually uses that protein to cause inhibition of the Wnt beta-catenin pathway, which causes neural development disruption. So how they did this is they actually took an adenovirus that was altered to include the encoding ROP, to include the gene encoding the ROP18 protein. And then neural stem cells were retrieved from mice, and those stem cells were transfected with that virus. And what you're seeing here is basically just the proof that the ROP protein was actually expressed in these neural crest cells, neural stem cells. Getting down to results, once they infected these mice, you can see here that the non-infected mice had a large amount of catenin. You can see over here too. They had a large amount of beta-catenin expression. When they used an adenovirus vector without the ROP18 protein, that actually decreased the amount just slightly. But once they infected it with the parasite protein, it actually significantly disrupted that beta-catenin expression. So then over here you can see the beta-tubulin. This is a microtubule component, a major microtubule component of the cytoskeleton. So they actually infected, this is the non-infected, and you can see this is a nice healthy cell, neural stem cell with the cytoskeleton. But once they were infected with that ROP18 vector, you can see how disrupted that cytoskeleton is. So this just proves more that this infection significantly disrupts the life cycle of that neural stem cell. So going even further, the neural stem cells actually transcribe these neurogenins, and these help to transcribe other factors that cause neural differentiation. So you can see here when they infected, this is the same kind of style, the non-infected, the blank vector, and the protein vector. So you can see in the non-infected, there was a significant amount of the neurogenin. We'll go over here so you can kind of quantify that. And then once you go to the infected ROP18 cell, you're actually significantly decreased. And this kind of follows with the beta-catenin from the previous slide. Beta-catenin leads to the expression of neurogenin 1 and 2. For our purposes, there really is no difference in the neurogenin 1 and 2, but this just shows another significant change in the expression of these neural stem cells. So what's the big picture here? This is my artwork, for lack of a better term, that I drew, kind of outlining that paper, and what they found was the mechanism of the neuronal abnormalities. So starting over, these are both neural stem cells. Starting over here, you can see that there's some Toxoplasma gondii that are being released from this vacuole. And as they're being released, the vacuole itself is releasing the ROP18 protein. So this can leave, and it actually attacks the Wnt ligand and causes it to not be able to land on the frizzled receptor. If you remember from before, frizzled actually causes the APC to break away from beta-catenin, allowing beta-catenin to land on the DNA for further transcription. But in this case, because the ROP18 is inhibiting that Wnt signaling, APC is holding on to beta-catenin, so it's not able to get into the nucleus to cause transcription. The neurogenin transcription factors are not able to be formed, and the whole cycle of the cell is disrupted. This, as I said before, this can cause issues with proliferation, differentiation, or it can also cause apoptosis of the cell itself. And this is just to illustrate again another vacuole with the parasite inside it. So these are my references. The only one I didn't reference was the picture that I drew here. That was from me. If you have any questions, I'm happy to answer them. Thank you guys for listening to my presentation.